Hello, this is Alice Formiga of eOrganic, and I'd like to welcome you to today's webinar on incorporating biological control of spider mites into tomato pest management programs. This is the final webinar in a series of 4 about biological control in organic vegetable production. The other webinars in this series are available on the eOrganic YouTube channel and in the eOrganic webinar archive. Today, I'm very pleased to welcome our 2 presenters. Dr. James Walgenbach is a professor and extension specialist in the Department of Entomology and Plant Pathology at North Carolina State University, located at the Mountain Horticultural Crops Research and Extension Center in Mills River, North Carolina. He conducts an applied research and extension program on management of arthropod pests on vegetables and tree fruits. Also with us is Dr. Tom Bilbo, who is a postdoctoral researcher in the Walgenbach lab. His research focuses on improving the biological control of spider mites and thrips in vegetable crops. So with that, I'm going to turn over the screen controls to Jim Walgenbach, who will be going first.
Thank you, Alice. If you can probably figure out that I'm Jim Walgenbach. I'm one with the gray hair that has any available here or any left. And I've been working on spider mites for about 20 years or so. And for about the last 10, it's been more kind of from a biological control perspective. And it's been a lot more enjoyable during that part than the first 10. But so what I'm going to do today, though, is go through a little bit about the biology of spider mites in terms of their pest status on tomatoes, some of the factors that contribute to mite outbreaks, some of the native biological control agents, and then just very quickly the discovery of Phytoseiulus Persimilis, which Tom is going to spend a good portion of the time talking about how to manipulate those populations for biological control in tomatoes. Okay, the two-spotted spider mite is one of the more serious pests of tomatoes, and I have a picture here, a close-up on the left, where you can see the two-spotted spider mite adults and the eggs. You can usually see those with your naked eye. I'll have to admit, though, I could see them a lot easier about 20 and 25 years ago than I can today. But in most instances, you can see the adults with your naked eye, usually on the underside of leaves. The eggs that I have shown there, they're going to be kind of difficult to tell or kind of notice. One thing I wanted to point out in this slide, once you get to the point where they're one of those mite species that can also produce webbing on the leaves. And it's usually not until you have very high populations that webbing appears. And if you get to that point where there is webbing, it's really difficult to knock populations back regardless of what you do, even with the best acaricides. So you don't want to let them get that far away. Two-spotted spider mites can damage tomatoes in one of 2 ways. They can indirectly damage the crop by when population is very large and just they're feeding on the foliage. It oftentimes causes early plant senescence, which obviously is going to reduce yields. And most of the yield reductions we see are due to fewer number of fruit rather than the size of the fruit. But when populations get large, they can also feed directly on the tomato and resulting in gold fleck, which you can see quite evident on the right-hand slide. So those are the 2 damage systems. So I put this in here to remind myself to point out that tomatoes are an ideal host for two-spotted spider mites for a couple of reasons. Number one, the production of the crop is such that it's oftentimes a monoculture with fairly high levels of fertility, and even organic systems, there's a fair amount of pesticides that often go into that system. And some of those pesticides can cause flare-ups of pest populations, and a lot of them also reduce natural enemy populations as well. This slide happens to be not too far from our research station. We're located in the Southern Appalachians in far western North Carolina, where tomatoes is a pretty common crop. Well, some of the challenges to managing spider mites on tomatoes. Number one, spider mites have a very short generation time, so they have a very high reproductive potential, can reach large numbers in relatively short periods of time. They also have a limited mobility. So what you end up with is populations that kind of stay in one area, And tomatoes, oftentimes, there's not a lot of rotation in organic systems because you have relatively small farms. Even in large farms, there's not a lot of rotation. And those populations become well adapted to tomatoes. Even though spider mites have a wide host range, it still takes them a little bit of time to adapt to an alternative host. Also, there's very few natural enemies on tomatoes. Tomatoes is a very toxic plant for a lot of insects, even natural enemies. So they have some due to the chemistry as well as trichomes on there, which when ruptured, they emit a sticky substance that is tough for insects to get around. And then they have a very low tolerance for damage. And I'll illustrate that in this next slide. Oh, before we go on to that, I wanted to point out how we go about sampling for two-spotted spider mites. They prefer to feed on the upper portion of the plant. So what we do when we're sampling is we take a first fully expanded leaf from the top of the plant. A lot of times that's about the third most recently expanded one. And we just sample the terminal leaflet on that leaf. So all we're looking at are 10 leaflets in 3 to 5 different locations in the field. So we go down in one location in the field, we'll sample 10 terminal leaflets off 10 plants in a row. And then we'll move around to some other place in the field. It's a pretty good system, works well for us. I put this slide on here to just show you the reproductive potential of spider mites on tomatoes and the low tolerance for damage. So shown on the black here, the line with the black line that goes way up there in the early part of August, that's the two-spotted spider mite population on a per leaf basis. So well over 100 per leaflet. So they can reach very high numbers. Then look at the bottom here, this red line. That's the economic injury level. That's at about 8 to 10 mites per leaflet. And once you get above that, the damage is such that it's equivalent to what it would cost to apply some type of pesticide to control it. So that, as I indicated, that's about 8 to 10 mites per leaflet. Then below that, we usually set the economic threshold to be a little bit below that economic injury level to give the grower time to react before that population exceeds the economic injury level. And that can range anywhere from about 2 to 4 mites per leaflet. So once you get 10 mites per leaflet or more, you really probably need to think about putting something on to control it. I also want to point out that there's a lot of pesticides that are put on tomatoes, and I've put some of the reactions in both conventional production as well as organic production. Pyrethroid insecticides and neonicotinoid insecticides can flare mite populations, as can some fungicides. And I'll talk about why that is in just a second. And then in organic productions, anything that has pyrethrins in it, such as pyganic, Azaria, those can also sometimes cause flares of spider mites as well. Things that control them or reduce populations, suppress them, there's many acaricides that can be pretty effective. And then the insecticide Radiant, which in the organic formulation is in Trust, that can also suppress populations. And sulfur is another good one, but although there's not too much sulfur put on tomatoes, and especially in warm weathering systems. Now I just put this in here to show you the how effective some of these acaricides registered for conventional use can be in suppressing populations. Obviously, the larger the number here, or the smaller the line here, the more effective it is. And there's some here that really, once that application was made at about day 2 or 3, it really does a good job of suppressing mite growth. But we don't have nearly the options available to us in organic production that are as effective as those are in conventional production. So it becomes a little bit more difficult. And here's some laboratory work we did where we just applied an acaricide at every generation. We applied these, you can see the different materials right here, Venerate, Grandivo, this Ghost neem is a neem oil application. Tritech, it used to be what's called safety side. It's an emulsified oil. Then I just put acromite in here as a conventional insecticide. And you can see that's the only one where the population goes extinct after applying it. All these others, you see an increase in population although it's leased with a kind of a high rate of Grandivo there. So, and Grebo does have some activity. In terms of biological control, there are a number of generalist predators that can have some activity. Coccinelles or lady beetles, anthochords and geocrids can be effective, such as this is the minute pirate bug, and then this is the big eye bug on the right here. Nabids, which I don't see too often on tomatoes. And then there are some native predatory mites or Phytoseiulus, which can also feed on two-spotted spider mite. But I'll tell you, none of these do that well on tomatoes. You're not going to see any of these do a great job of reducing populations in tomato systems. But one thing that is very effective is a naturally occurring fungal pathogen of spider mites, Neozygotes floridana. This is native occurring, and it can decimate a population during very humid conditions. If you get a rain event, it can just decimate populations. But once that weather dries out, the pathogen is no longer really active, and populations can just increase considerably. Now, one thing to note, though, is, for instance, on conventional grown tomatoes, the fungicides placed on that essentially eliminate this as a biocontrol agent. But it is kind of interesting how if you can go around a tomato field, there'll be tons of mites in there, and you can go outside around that field in the weeds and things, and you won't see any. And a lot of that's due to this pathogen. I wanted to point out, this is kind of leading into some work with Phytoseiulus Persimilis, which we'll get to here now. This was some sampling we do. We do experiments oftentimes, and a lot of it is on farm with growers in two different areas of North Carolina. One is in the mountains, this Henderson County here, which is closely related. As a matter of fact, the red dot there is where our research station is. And then in the Piedmont location, this county is called Rowan County. They also have quite a bit of vegetable production in that area. So we do a lot of work there. And it's probably about 12 to 15 years ago, we first saw this predator that was occurring on tomatoes. It was usually kind of the mid or latter part of the season. It was down in Rowan County. Eventually, we had it identified as Phytoseiulus Persimilis. First thing we did is do some overwintering studies where we put some winter weeds, infested them with spider mites, and then put the predators on there. We planted them down and planted them under these sheltered areas, both in the mountains and down in the Piedmont area. And we were able to detect them throughout the course of the season in the Piedmont area. They didn't do too well in the mountains. It's a little bit too cold up here for them. But they do survive year-round down in the Rowan County in the Piedmont area. One thing I wanted to also emphasize with this predator, it's a specialist. It only feeds on spider mites. So if the spider mites go extinct, so does the predator. It's a voracious feeder of mites. It can eat up to 20 eggs or 10 adults per day. And it's well adapted to high temperatures and high humidity. And it's actually used quite a bit. It's the most commonly sold natural enemy by insectaries throughout the world. And they're used frequently in California strawberries and in greenhouses as well. So I wanted to show you the life cycle of both the two-spotted spider mite and Phytoseiulus persimilis is identical. They both have an egg and then then a larva and two different nymphal stages before they molt to the adult stage. Now, just look underneath here, though. This is the number of days it takes to complete development from the egg to the adult. For instance, at all these temperatures, it takes longer for the two-spotted spider mite to complete development than it does the predatory mite, which is a good thing because the spider mite is reproducing at a greater level, and it's going to outproduce the two-spotted spider mite. Now, it's offset a little bit by the fact that spider mites do produce more eggs. And this I put in here just to show you typically what we oftentimes see in this Rowan County area, where early in the season, these dots here represent when an acaricide was applied. These the blue line is the two-spidered spidermite population. The red line is Phytoseiulus. So in those early plantings, rarely do we see Phytocelius Persimilis show up until later in that crop. It's usually too late to really have much of an impact. But it's around early enough for those mid plantings of tomatoes to be controlled quite well by Phytoseiulus. And that's usually in August or into September. Now, this is the third planting. You'll notice it wasn't controlled that well. And the reason for that is because that particular crop was planted in a more isolated area where the predator was not present. So oftentimes, if you have these plantings sequentially planted next to each other, it's more easy to get continuous biological control. Once we did that, we also did some rearing of this in the greenhouse. And to do that, now Tom will expand on this a little bit more. This was just where we had a greenhouse where we reared two-spotted spider mites, grew the tomato plants, then grew mites on them. And then we moved them to a different house where we put Phytoseiulus on there to grow the Phytoseiulus. Then we did some work where we looked to see how well it controlled the spider mites in the field. And I guess the only thing I want to point out here is this two-spotted spider mite cumulative mite days. The higher the number, the higher the population. You can see where we had no predator release and no acaricide, we had the highest number of spider mites. The lowest is where we had both a predator release and an acaricide. But even where we just did the acaricide, we had lower mite populations, although not significantly, than where the acaricide was applied with no mite. So it showed us that these predators can have an impact on two-spotted spider mite population. They can do a good job of controlling it. Unfortunately, if you put the pencil to the paper and figure out the cost of it, if we release them at 20,000 Persimilis per acre, and we put it out in the whole acre, it's going to cost about $741, which just doesn't match with any type of acaricide. So obviously, we need to do something different to make this a little bit more affordable. And that's where I'm going to hand it off to Tom now, and he's going to talk about some of the work he's done to manipulate populations to control pests in the field.
Okay, thanks, Jim, for the intro, and thanks for everyone for tuning in. Yeah, so like Jim said, where we're at now is we're trying to improve this system so that it's better economically and has more reliability, so that this can then be a practice that is more adopted by growers. And I'll be talking about conserving those predators that are showing up naturally, releasing them ourselves, and also how you can rear your own populations to release into the field. So like Jim said, we have these naturalized Persimilis populations that they reoccur yearly in the Piedmont and down in the coast as well, but not up in the mountains. And they can do a pretty good job, but it's spotty, and sometimes they show up too late to do much. So one of the knowledge gaps for improving the pest control provided by the naturalized populations is to get a better understanding of where they are in that overwintering period between the growing seasons. It's sort of a black box right now. We don't know where they are and what factors decide those distributions. So we want to know if we can enhance these natural populations to make them show up earlier and to be more widespread. So, you can picture them, they're in these fields as the season goes on. And then at some point, it gets in the fall, a frost comes through, all those vegetables are done with. Those Persimilis, they need spider mites, they disperse, but where do they go? So like Jim had said, these populations are obligate specialist predators. So we only expect them to occur where spider mites are occurring. Also, Persimilis can't enter diapause, so they don't have the capability to just hunker down into a dormant state. They need to have some degree of activity, and they're going to be where the spider mites are, we presume. So, we want to know where they're going after the vegetable crops, where they're overwintering, what factors are deciding where they are, both spatially and in time, and can we do anything to manipulate the habitat to enhance the predators? So one of the things we first started going out is, can we go out and can we find them on these these plants that are out throughout the winter. They can host spider mites, many of them can. So, which ones may host Persimilis, and therefore that would help us get a better understanding of at least the plant habitat that would be around harboring these populations. So we'd go out, we looked at all these plants visually. We also collected samples, both plant samples and leaf litter samples around these plants, and we would put them through Brulesi funnels, try to find them that way. And long story short on trying to go out and find them is that we know the Persimilis predators are out there, but they're pretty dispersed, and it's a low probability. And so it's a bit like finding a needle in a haystack. So we did all this sampling and pretty much turned up no Persimilis whatsoever. So we had to try some other strategies. Something else to consider is that there's a lot of strawberries grown in these areas that vegetables are grown in. And strawberries, they're there all winter. They're a good spider mite host. And so we thought we would look in these to see how Persimilis do and if they show up in strawberries. And also, instead of going out and trying to look for the Persimilis, can we attract them and bring them to us? So that's where we tried out these sentinel plants. If you can see my cursor, they are these mint plants that are tomato, two-spotted mite infested, and they're highly attractive to Persimilis. So can we put them in areas and detect Persimilis in those local areas? So some of the early results we have with the sentinel mint plants, by putting them out around tomato fields, we put them out around tomato fields that, the tomatoes maybe were there, they were late planted, which means they would be there all the way up until the frost, or mid-planting. So they would end a little bit earlier, and there'd be a little bit more of a gap between when the vegetables were there and then when winter kicked in. And it seems like from the early results that the later those tomatoes are there, and hence the spider mites and the Persimilis, the more likely they are to still be in that localized area. That's what we had found from some work last winter. So basically, they haven't had a chance to disperse away yet, because once they go through fields and they eliminate spider mites, they need to find more spider mites. And in strawberry fields, like I said, they will oftentimes have spider mites in them throughout the winter. So we put Persimilis in one strawberry field, and we were able to pick it up every single month in the winter, which is a good indicator showing us that Persimilis can do well surviving in strawberry fields throughout the duration of the winter period. So we know that this could potentially be a bridge between the fall and the spring where Persimilis could be. So that makes you wonder if we could modify the habitat to attract Persimilis. So we know those mint plants are highly attractive. They seem to do a pretty good job of attracting Persimilis. Instead of just having a couple in order to detect them, could we do small plots of mint, infest them with spider mites, have those plots attract the Persimilis there, build up the numbers, and then when those early tomatoes or other vegetables are nearby, those Persimilis can move right on over. So that's something that we're going to be trying out this spring to see how effective that might be. So switching gears a little bit to releasing predators ourselves. So these would be ones that we either buy or we're rearing our own in lab. And, is this a way that we can get good spider mite control that's more reliable and earlier in the season? So whenever you're going to release predators into a field, you have to ask yourself a couple questions. You have to know, where are you going to put them in the field? How many are you going to put out? When are you going to put them out? What's going on in those fields around you, and how might that impact what your predators will do? And then, for some instances, like with Persimilis, it could be you may need to know the strain of those Persimilis, whether they were pre-adapted on tomato or a different plant like bean. I won't talk about that much here, but if you're curious about it, definitely ask a question and I can elaborate on those strains. So through a number of trial and error throughout the last previous years, we've found out what works pretty well is that we'll release Persimilis at a rate of 20,000 per acre. And we'll do those when the spider mites first enter and show up in the fields. As soon as you see them there, they have to have food in the field or else they'll just disperse away. We would do this in a small section of the field, about 5 or 10%. As Jim had said, doing a release in the whole field is not economical, and importantly, what I'll show here in a second, not necessary. And then we'll go and sample our spider mites and Persimilis. So these Persimilis, you can rear them yourselves. We do this in-house. This is typically how we release them into the field. You can also purchase them from a number of suppliers. And there may be some differences between how effective these are, but we have had success with both in the field. And then you go out in the field. This is always the fun part. You go out and you count spider mites, count Persimilis, and you see what's going on every week. And if you're wondering what that looks like, it's always convenient to have a hand lens that gives you 10x magnification. These are very tiny mites we're dealing with, but with a hand lens, you can see them pretty well. So I'm going to go over a little bit of, just a little bit of the data we have to illustrate how effective Persimilis can be in managing spider mites. Throughout the last 2 years in the Piedmont, we've sampled 21 commercial tomato fields. We released predators in 8 of those, and we detected naturalized populations that naturally occurred in 4 fields, but in only 2 of those did they show up in time to suppress spider mites. Oftentimes they can show up too late. So here's one example. This would be a tomato field that is about 3 acres in size, and it's split into 11 sections, split by drive rows, which go up and down fields. So if we take our Persimilis and we release 2,500 into section 2, and then we go out and we sample weekly and we see, what happens. This figure, all it's showing is it's showing the number of spider mites in black and the number of Persimilis in red over time. That's about a couple months. And so you see the spider mites, they get in the field, they start to build. We put the Persimilis in there. They continue building. They had a minor little setback with the miticide, but they didn't get too phased by it in this case. And they just kept taking off, exceeded the threshold, but those Persimilis really expanded in abundance and just completely took them out after about 5 weeks. And this was a case where there'd be local extinction. So the spider mites basically eliminated from the field, and then the Persimilis followed because they had no food. So that was the entire field, but it can help to understand what's going on by breaking it down into sections, because these Persimilis, obviously, they did a good job of managing the spider mites. We released them, and then they quickly dispersed throughout the whole field. So just to break it down by section, these are just showing that same data I just showed you, but from the release section at the top to the far end of the field at the bottom. And you can see in the release section that the spider mites stayed low the whole time. They never exceeded the threshold. And Persimilis took them out just fine. But there was a little bit of a delay at the far end of the field, but they, field-wide, they still wiped out the spider mites. Just a little bit of delay at the other side. So it's almost like a wave moving across the field of predators. And then this is pretty typical of what we'll see, is that we release these Persimilis, and then 3 weeks later, you can first detect them. I mean, they're out there doing something, but they don't really become abundant enough to detect them by just random sampling until about 3 weeks. At 4 weeks, they've really increased in numbers. And then you start to see the decreases in spider mites. And by 5 weeks, you typically have eliminated spider mites from the field. So they quickly disperse throughout the whole field. But there are remaining questions about how to improve this system. We don't know if this field was bigger, the extent of how far they would disperse, and the extent of how far away would spider mites have been managed. We also don't, so that's with the field size, influencing this dispersal. There is an impact if you have surrounding crops with spider mites in them, because it will draw Persimilis basically to them to some extent. I can talk more about this if you're curious. And then miticide applications might influence dispersal. We've seen possibly some evidence for this, but we're not sure. And then another big question is how to make this system more economical, because that's really the key here. We know they can be really good. We want to make them economical. So one idea is to use sequential releases of Persimilis across multiple plantings. And this can be, when you put Persimilis, like I showed, in a field, they increase to very high amounts. And so basically that field becomes a little Persimilis producing factory. So when those Persimilis are now in huge numbers, hundreds of thousands in the field probably, you can then take those Persimilis and then you could start moving them to other fields. Or consequently, if you had two fields that were staggered in time next to each other, you could have the same effect. And so this is an example that illustrates this well. This was a 7-acre field in total, and it was a June planting right next to a July planting. So in that June planting where the tomatoes were first, as soon as spider mites showed up, just like last time, we had our Persimilis in one section. And then we would let them move across the field through the June planting and then into the July planting in time of spider mites showing up. So on the left is that June planting. And you can see the spider mite and the Persimilis trends again with the black and the red, spider mites in black. You see, just like last time, Persimilis, the spider mites get in there, they start to take off. We got the Persimilis in there and they just completely wipe them out, local extinction. But then you got these two plantings next to each other. Persimilis it seems that they just moved right on over into the July planting and they kept spider mites extremely low the entire time. And you can see that action threshold up at the top. That's way up at the top of the figure. So having these plantings next to each other, you really get the benefits of releasing them in one crop across two crops. And if you don't have plantings next to each other, you could, in theory, we're going to test this out this more this summer, but collect a bunch of leaf tissue with Persimilis on it, move it into other fields. So that will greatly expand the cost effectiveness of this method. So, Jim alluded to the fact that spider mite numbers can get extremely high in some of these tomato fields, even when you're trying to manage them with miticides. These are two examples of fields that didn't have any predators, and you can see that the spider mites get in there, they take off. You can knock them back a bit with miticides, but then they just explode, and the numbers are huge, and you get a lot of spider mite webbing and a lot of damage to the plants and the fruit. And this is just two examples here. A couple miticide applications kept them down, but as soon as those were gone, spider mites just exploded. So you might be saying, if we can keep the spider mites down until we're pretty much on harvesting, who cares? Another thing to consider, though, is that these insects will move from one field to another to some extent. And so if you have a system without Persimilis and those spider mite numbers are building a lot, once that field's done, those spider mites very well will move into surrounding fields. So you actually are creating a problem from one field, and it can become a problem for another field if the spider mites are exploding. But if you have Persimilis in there, those numbers are going to be approaching 0 spider mites. And so you basically go from having a population source to a population sink. And that's another benefit here to think about with this biocontrol system. So because we've had a lot of these releases, we've learned a lot from successes and even more so from our failures. There's this principle that comes up every now and then called the Karenina principle. It has a great opening line of the novel by Tolstoy that all happy families are alike. Each unhappy family is unhappy in its own way. And this principle is just, people use it to illustrate that a factory, you basically need everything to go right for it to succeed, but you could have a failure in any single area, and that will result in the whole endeavor failing. And so we've seen Persimilis releases not work for a few different reasons. One, this one may be the most common, is that you don't get Persimilis in there early enough. You really want to get them in the field basically as soon as spider mites are showing up, because they take a couple weeks to build. You could also spray pesticides that are harmful to them. This goes for both organic and conventional. Any of the broad spectrums like pyrethrins can sometimes take out the Persimilis. Extreme weather events, we've seen this setback releases before, where you get Persimilis out and then a huge thunderstorm can come through and just wash out the field, and that can really disrupt things. And this last one's a bit tricky, where if those plants where you release Persimilis were struggling for whatever reason, they got set back because of water stress or disease, this can actually have a significant impact on how good of a Persimilis factory that area is. And so if the plants are not doing well, the Persimilis won't do well. So like I said, we rear our own Persimilis, and it's not too difficult to do. And we're going to be publishing something on our website here pretty soon outlining how to do this in more detail. But the basic premise is that you have to have two separate areas. This is what's important. You have to have one area for rearing spider mites because Persimilis are specialist predators, and then another area for rearing Persimilis. And you really got to keep them separate. You think rearing Persimilis might be the hard part, but actually keeping Persimilis away from your spider mite colony can be pretty tricky, and it takes a lot of good sanitation. You never move around between the two. And so the basic premise of doing this is that you get all these tomatoes going when they're about 5 inches tall. You would put a bunch of spider mites on them, and then give them about 4 to 5 weeks to really build up. And as soon as you start to see webbing at the top, that's a good sign to introduce Persimilis. Depending on how many plants you have, depends on how many you would add, but usually just a couple hundred. And then you let those Persimilis build up in there for 3 weeks. Usually you can count, and then you would have to look at count them on the plants. I take 3 out there, count to get an average, just to give you an idea of how many you've produced and therefore how many you need to put out in the field. But we have been able to produce a lot on a small scale. That really only takes one person, not a lot of space. And so, for example, one batch, if we had 360 plants, 20 of these 18 cell trays, we've been able to produce up to 100,000 Persimilis in this system. And so if you're only releasing in 10% of a field, this would give you potentially 50 acres of tomatoes, which is pretty good. So this system works pretty well. Once you get it down, it's not too difficult either. And, as always, with any biocontrol agent, if you want your biocontrol to do well, you have to avoid using certain pesticides. This is maybe a bit more of an issue in conventional than organic, but there are still organic insecticides you got to be careful with. We have a chart published on our website that lists a whole bunch of insecticides, conventional and organic, and it shows you the toxicity to a few different predators. Persimilis here on the right, and that minute pirate bug, which is a really good thrips predator in peppers and other crops there. So that's just an example. You can go to the website and find this chart we have. We have a whole page on there about Persimilis, describing a lot of this work further. So yeah, definitely go and check that out. And then a big thanks to everyone that's part of this, and all the funding sources is huge. And the lab, and I'll leave you with this pumpkin.
So starting at the top here, there are studies that show that sulfur can increase the two-spotted spider mite populations as well. Any thoughts?
I've heard of that in the past, but in general, my experience with sulfur is that it does suppress spider mite populations. And in fact, that's a pretty common treatment on peaches a lot of times, especially organic peaches, is sulfur. And I'm not too familiar with instances where sulfur increases populations. The only thing I can possibly think of is it was a really warm, hot environment where the sulfur is probably going to have less of is going to be doing more harm to the plant than it is to the pest. But no, I'm not. I would tend not to agree with that.
All right, thank you. Are you using a rotation of those insecticides that you were talking about, Jim? And also, how many days after the release of the predatory mites are you applying the miticides?
Well, I'll let Tom answer that. He's done that. Usually, those miticides are applied not too long after we've released the acaricide. It's usually if you have a really large population, if you use the right acaricide, you can apply that in advance and help get things into a balance a little bit more, so that the predators suppress the population, so predators have a better chance. And maybe Tom can answer that more.
Yeah, I'll just say, at least in the conventional fields, since the vegetables are so labor intensive, the miticide use is sometimes dictated by when they're going to be harvesting or when they need people to be in the field, because you have restrictions on re-entry intervals and post-harvest intervals. So that dictates some of the miticide use. And we were doing these studies in conventional fields where there was a lot of spider mites, so they were a good example. And we didn't necessarily want to interfere with a lot of the things the growers were doing, so we would often just say, do what you're ever going to do. We can't guarantee the Persimilis are going to work. It's still experimental. So they would go out there and do what they thought they needed to do, regardless of the Persimilis. But like some of those data slides I showed, the miticides can help. Sometimes they work better than others, depending on if there's resistance or not.
I will just expand on that.
Yeah.
These Persimilis, they're pretty tough individuals. They can tolerate a lot of different pesticides. Now, if you get to some of these other things like really broad spectrum things like pyrethroids and things of that nature, it's gonna take them out. But it's pretty tough to get rid of them once you have them out in the field with pesticides. And some of the caricides, usually, fortunately, the ones we work with are pretty selective, and they have very low toxicity to the predator, to Phytoseiulus as well. So.
Yeah, just to briefly add, I mean, those slides that I showed had miticides in there, but there's a bunch of other things being sprayed for stink bugs and caterpillars. So the Persimilis, yeah, they succeeded despite a lot being thrown at them.
Okay. Were the instructions that I believe it was Tom was talking about on how to apply the Persimilis, would that apply to a greenhouse as well? How would you adjust the application for a greenhouse?
The greenhouse Persimilis have been used in greenhouses since the 80s. That's probably how they got established in the area. They probably escaped from greenhouses, or maybe if people put them out in strawberries or something. But my understanding is that there's well-established rates in greenhouses. The dynamics in there are going to be different. And I think all those companies that sell them, they give you very specific rates for different crops about what you should be doing in a greenhouse system. So the novelty of what we're doing is that they're not really used. The only field situations that Persimilis have really been used in are strawberries in California and Florida. And that's sort of, I think, it. We're really trying to just show that this can work in field tomatoes under field conditions.
I should also point out, some of these growers we're working with, they're growing a fair amount of hemp in greenhouses as well. And I tell you, spider mites can really do a number on hemp, especially once you bring them in the greenhouse, they just go crazy. And several of these growers have controlled spider mites on hemp in the greenhouse exclusively with Phytoseiulus Persimilis. Now, the whole key there is to make sure the spider mite population isn't too high. Again, the whole key there is to, as soon as you see spider mites, you want to get your predators in there. If you start to see webbing, it's tough to get things done quickly.
Yeah, to draw that distinction, waiting until webbing is good if you're trying to produce the optimal amount of Persimilis in your own rearing program. In the field or in the greenhouse, if the webbing's already there, that is too late. So important difference. If you want to produce the maximum amount of Persimilis, you don't care how much damage is done to the plants, wait until you just start to see that webbing. But don't do that otherwise.
So that really affects the timing of the application as well.
Mm-hmm.
Okay, let's see. Where is Persimilis coming from if they're naturally there but showing up too late to control the TSSM?
Well, the reason they're showing up too late is because during the overwintering process, both two-spotted spidermite and the Persimilis populations, there's a pretty good level of mortality that's occurring over that period of time. So what you really see is spider mites entering tomato fields, and then there's a lag time between when the spider mites build up to large enough populations and Phytoseiulus Persimilis really detects them. Usually you don't, in nature, Phytoseiulus is not going to show up or is not going to be attracted to a spider mite population unless it's pretty high. They're giving off chemical, or there's some chemicals involved in that and other things. But So what it is, it's kind of a lag time in terms of the predator has to build up to large enough numbers to be able to be detect in our sampling program, and the spider mite population has to be large enough in order for them to be attractive to Persimmons. So that's why we see oftentimes they show up what we think is too late in tomatoes. They're early planted tomatoes. I'm not sure if I was clear on that.
Okay. Were the data counts that Tom presented also from terminal leaf samples?
Mm-hmm.
A question about that.
Yeah, we do all this sampling the same for at least in our lab.
Okay. Let's see. I'm trying to read the question here. Do adjacent crops attract Persimilis differently depending on what host they were raised on?
As far as I mean, that's a good question. I don't think I would say we don't know. The point I alluded to before about how the surrounding crop might be influencing what's going on in the crop, that if you have a tomato field and you want to put Persimilis in there, well, we realized we had put them in one of our fields, we put them in there, but there was another tomato field nearby that it's kind of dumb, we just didn't really factor it in, that had a lot of spider mites in it because that's what they do. And it definitely pulled it, it's basically pulled the Persimilis in two directions. And so the response of Persimilis ended up being too diffuse, and we didn't really get good management in that situation. So that's why, if you're going to be putting them in fields, especially if there's other fields nearby that have spider mites, it's food for Persimilis. They're going to want to go there. You'd want to maybe, that would decide where you're putting them into the field. So if you're only going to put them in one group, it should be where they basically only have one way to go. So you know that you'll at least get control to some extent as they sort of, move like a wave.
I think the most attractive things to spider mites, the most attractive things to Persimilis are spider mites. That overrides plant effects, I think.
Okay. Yeah, we have a couple of questions about whether they also eat other types of mites, such as red spider mites.
It depends what red spider mite is.
Okay.
There's some morphs of red spider mites that it's there's an argument whether it's a different species from two-spotted spider mite or the same species. Those ones they will eat, but they're pretty specific. They will not eat, for instance, European red mite on tree fruits. It's got to be the two-spotted spider mite.
Okay. Would you expect that this predator would also be effective in the Pacific Northwest region, especially in protected cultures, like in greenhouse high tunnels? And is it also effective on mites on cucumbers?
Yes, I've seen in the same area down in Rowan County where we do a lot of our work on tomatoes that there's cucumbers and watermelons growing down there, and I've seen Phytoseiulus on both of those crops. And they get in there, they'll take the spider mite population out fairly quickly. So they will go to cucumbers, and I would expect them to be effective in the Pacific Northwest. It's a humid environment. Well, depending upon where you are in the Pacific Northwest, it's a pretty diverse environment. But anytime you have high tunnels, that's going to increase the humidity. But yeah, I see no reason for them not to be effective in the Pacific Northwest as well.
Okay, we have a comment that says Persimmons doesn't do well in the Pacific Northwest where RH is low. I'm not sure. What does RH stand for?
Relative humidity.
Oh, relative humidity. Okay. No.
Humidity is a really important determining factor for how well they can reproduce. A lot of the predatory mites, I believe it applies to most of them, is that the like the egg hatching and everything is much more successful once you need to get above like 50 or 60% humidity. It doesn't need to be consistent. I think there'd been some studies showing that you could have maybe a lower humidity during the day, but as long as at nighttime the humidity gets up, at least if you have to have at least some period of high humidity to ensure that there was a good reproductive success and survival of the predatory mites like Persimilis. So the humidity is really important.
Okay, thank you. That's great. Then we have another question like that from the Midwest. There's a comment that they've had a few spider mites in tomatoes, but not bad. But under dry conditions, they expect huge increases. The weather is trending to dry, hot summers, and that suggests we will have issues potentially in commercial organic tomatoes. Says cold temperatures might wipe out the good guys.
I suspect in the Midwest, the winter temperatures are definitely going to knock out the good guys. But if the nice thing is there's multiple sources where you can buy these spider mites or buy these predators during the course of the year, and you can get them in a pretty quick period of time. What is it, Tom? Maybe less than a week.
Yeah, they overnight them 1 or 2 days. Some companies only sell them on certain days of the week, depending on how they produce. But you can get them within a week, and depending on when they're producing them to sell.
So really, the best option there is if you anticipate, if you notice spider mites and you anticipate some drier weather, it's not too late to order them and get them out there.
Okay, great. This question about spider mite dispersal. Do they tend to invade fields from the edges, or do they have a more random initial distribution?
I presume that's for when we detect the naturalized ones, we're going out in tomato fields, we are sampling the whole sort of randomly throughout the whole field for spider mites. And then sometimes these naturalized populations will come in, and sometimes it seems random. Part of it is the sampling error, like there's just sort of the probability of when we might pick them up if they're at low numbers. So we pick them up in different parts of the field. That's my experience the last 2 summers working on it. They can kind of, you don't know. And again, the spider mites are not necessarily even. Sometimes they are not equally dispersed throughout a field. So wherever the spider mites are strongest should be the most attractive area for Persimilis to be brought there.
It gets a little complicated. I think generally they do come in from the edge of the fields, but tomatoes are a crop where there's so much hand labor that goes on in there. At least we're all dealing with staking ones, and so they're getting moved around by the farm workers a lot within the field. So initially, they might appear to be on the edge of the field, but after a while, I think they're just getting moved around by workers throughout the whole field, so you really don't detect a normal pattern of invasion from one edge to the interior.
Okay. Let's see. Here's someone who had a greenhouse full of tomatoes, and spider mites are their #1 problem. They're in Southern California, and they had webbing and ended up discarding all the plants. So I guess the question, she wants to know when to apply the Persimilis this season or the miticides. But I mean, I guess, can you describe the process of how one would go about scouting for spider mites, and at what point you would then apply the Persimilis?
Well, I'll let Tom expand on this a little bit more, but if it's California, Southern California, I think she said,
Yeah.
it's going to be hot, and if you had a problem where it was so bad you were getting webbing, you can probably guarantee you're going to have a repeat problem again. And my advice would be to, as soon as you detect spider mites in there, I would, as soon as those plants are big enough and you see the first spider mite, I would order some predators and get them in there very early in the process. Tom, what's your suggestion on that?
Yeah, ideally, if I had my own operation, I would try to keep track of if they were showing up, if they tended to show up at the same time. We're maybe working on developing a model to better predict when they arrive in these fields. But if they're showing up first is this, try to find out if keep good records, and you could just see if they're showing up at the same time. Then you could maybe be a little predictive about it. For us, we go down to these field sites. They're 2 hours away. We go down once a week. And if we pick them up 1 week, so in the Piedmont of North Carolina, we're talking like in the summer, low 90s, upper 80s, a lot of times in the day. As soon as we detect them 1 week, we know that when we go back down the next week, we need to bring predators with us to release, or else it'll be too late.
Yeah, and a good point. I don't know if you can remember, spider mite growth is almost like logarithmic. It can be very low for a couple of weeks, then all of a sudden it's got the appearance of just zooming up there on a linear rate. It takes them a while to get established, but once they're established, boom, they really go to town quick.
Okay. Do any of the growers that you work with use things like Azaria fumoso rosea or Bovaria bassiana or other bioinsecticides in combination with Persimilis to successfully manage spider mites?
The Azaria can be kind of tough on them. That's got a pyrethrum in it, same active ingredient as pyganic. And another organic thing that they do not like is surround or kaolin. Predators do not like that, although we don't see that really used on vegetables at all. It's just too difficult to wash off, and then you got that all over the fruit and everything. But and that's an interesting one in terms of Bavaria bassiani. Foliar applications, I'm not sure how serious. You would think it's going to have an effect knocking them back somewhat, but I really don't have a good sense of the Bavaria. I think I've noted. Tom, have you ever seen any publications about Bavaria bassiani effect on predatory mites?
Some have been done, but I don't recall what the results were. Just quick note. Jim, you responded to that being it was another bioinsecticide. I think you were describing, just to clarify, Azaria insecticide versus the biologicals.
Let's see. Will the fungal pathogen that you mentioned that's toxic to the two-spotted spider mite persist in the field if humidity remains high? And if yes, at what percentage humidity will it thrive?
I can't give you a good percentage. Definitely well over 60% humidity. And even in periods where there is, it can be humid, which it often is in the southeastern United States here. It's humid all the time in the summer. That by itself is not going to be sufficient for these epizootics to take hold. It usually takes a rain event in order for that to really have a big impact. Humidity alone generally is not that effective. You need to have it associated with a rain effect.
Okay, thank you. If the population of spider mites is already high, where in a greenhouse, would you recommend knocking it down with a miticide first and then applying the Persimilis?
I would, yes.
Okay, thank you.
You have to be selective as to which miticide you're going to use.
Okay, and you have more information on your website about which insecticides are toxic to the Persimilis. So that sounds like a really great resource, which we can post along with the archived webinar on the eOrganic site. Thank you all for asking these questions. It looks like we've reached the end of the questions in the queue here. And I'd really like to thank you, Tom and Jim, for this really informative webinar, and I look forward to hearing more about what comes next in this work and seeing some of those published papers. So thanks again, Tom and Jim, and thanks to everybody for joining us today.
Thank you.
Yeah. Thanks so much for tuning in.